Okay, when we consider cell, actually cell are considered to be the structural and functional unit of all life form. Cell is the structural and functional unit of all life form actually. Okay, I will quickly help you with the revision portion, you don't worry. When we consider cell in our study, we start with its meaning and its from where it has been originated. It has been originated from a Latin word actually. This is a Latin word, I should say, and this Latin word cell actually means a little room. That means cell is something that has a meaning as a little room and everything arranged in it. Got that? Okay. And this cell is actually the fundamental unit of all life form. Whatever the life form you observe here on this earth, all these life forms are actually because of this cell. That is why we call it fundamental unit of life. And we define cell as the structural and functional unit of all life form. Here I'm here with this chapter. While we now the question arises, how have we just because it was not invented, I should say it was discovered. Okay, in previous class I have just told you that what is difference in these two words, discovery and invention. There are two words, one is discovery, another is invention. Okay, if we consider a cell, what will you call cell is a discovery or invention? Tell me, Sanket. What do you call to cell? It was discovered or it was invented? Which word we need to use and why? Yeah, exactly. Cell was actually discovered. Why do we say so that it was discovered? Because, yeah, exactly. Discovery means we are just going to remove the cover out of that. Okay, we are not able to see that thing because there is a cover. And if we are removing the cover, we can just see that. Actually, cell was there earlier, and it was Robert Hooke who first observed this. That means Robert Hooke has discovered the cell. But if we consider electric bulb as an example, whether you call it discovery or invention, yeah, exactly. Electric bulb is said to be invented because it was not earlier present, and it was Thomas Alva Edison who first properly manufactured, created it, or you should say properly invented this. It was invented by the scientist uh, Thomas Alva Edison. Actually, he has experimented 1,000 times. Then only he could invent this electric bulb. That is why we need to learn from his perseverance, his patience. Being patient about anything always give you a result. Uh, you keep on working, and that will improve your that will improve your uh, skill, or that will also improve your learning, and once you will be able to gain that you want, or you will be able to achieve that you want. That is why it is suggested that being continuous is very much needed. Being patient is very much needed. Patience is very essential tool that we have, that we should apply there in our life, there in our study. Okay, the fundamental unit of life is actually a cell that we are studying today, and this cell was first observed, or I should say discovered, by the scientist Robert Hooke in 1665. I want you to read this paragraph very carefully, and you will be able to understand how it was discovered. while examining a thin slice of cork. When he was examining this cork, Robert Hooke saw that cork resembled the structure of a honeycomb. It was quite like honeycomb structure, consisting of many little compartments. And because there were many little compartments in it, cork is a substance which comes from the bark of a tree. Do you know what is bark of tree? From that bark of tree, we get this slice of cork, and when Robert Hooke saw that cork, it resembled as the structure of honeycomb, which has many little compartments. Uh, that is why it was named as cell, because cell actually has the meaning a little room in Latin, and that is why it was given a name cell that time. This was in year 1665 when Hooke made this chance observation through a self-designed microscope. He has actually self-designed the microscope by using which he could discover this cell. This may seem to be a very small and insignificant incident, but it is very important in the history of science. This was the very first time that someone had observed that living things appear to consist of separate units. The use of the word cell to describe these units is used till that day in biology. Let us find out about cell. We will just learn what these cells are. What are living organisms made up of? Now, if someone asks what does these living organisms have, what are living organisms made up of, then our answer is going to be they are actually made up of these small units known as cell. It looks like these little rooms, and that is why we uh, started using the name cell at that time. And there is one experiment related with the cell that we can just go through. Then the question arises: what are these structures? Then we find that these activity, that onion bulb of different sizes have similar small structure visible under a microscope. We can use this uh, onion uh, peel experiment to observe this cell. But all organisms that we observe around are made up of cells. However, there are also single cells that live on their own. Okay, that is why we can divide them as a single cell organism that are unicellular, and many cell organism that are multicellular, that I have already told you in previous class. I will just tell you all these things once again. Okay, this is basic unit. Inside the cell there are different parts performing different activities to keep the cell alive and functional. These are cell organelle, and there are different cells that we are going to study there in our syllabus, or rather we are going to uh, focus on later on in our study as well. Like nerve cell is there, muscle cells are there, from which we can say that our muscles and our skins are made up of. Then bone cells are there, gland cells are there, blood cells are there, and for reproductive cells we have two kind of cells. In case of male, sperm is a cell. In case of female, ovum is a cell. Even fat cells are there. A lot of cells are there, but I want you to remember these 8 cells that I have presented in front of you. After this, we can focus on some of the scientists, some of very interesting scientists. Okay, I have one more slide for you to go through about these scientists that I have promised you that I will help you with those scientists, and I have that entire slide in which I have presented those important scientists in detail. Okay, I'm just telling about those scientists. The first scientist is Leeuwenhoek, that I want you to focus on, and I have, yeah, here I have. The first scientist is Robert Hooke, obviously. The cell was first discovered and named by Robert Hooke in 1665, and when Robert Hooke observed this by using their own microscope, he found that it looks like honeycomb, and these are very small apartment, like a small apartment. That's why he named it cell, which actually means a little room. It is his book in which he just explained all these things. And actually, Hooke saw was the dead cell walls of plant cell. He used cork that I have already told you. From bark of the tree we get this cork, and from this cork he saw all these things. Then I have another scientist. We have another scientist, Antony von Leeuwenhoek, very famous scientist. He was the first biologist who observed the bacteria and protozoa. That means he first observed microorganisms, I should say. Leeuwenhoek made microscopes consisting of a single high quality lens of very short focal length. It was quite like this. He used the microscope like this, and he first observed bacteria and protozoa. Then we have another scientist, Purkinje. Purkinje Dujardin is one more scientist, and Purkinje is another scientist. Actually, Dujardin discovered the protoplasm and named as Charcot. Further, J. Purkinje first introduced the term protoplasm. First, this term was introduced by Purkinje. Protoplasm is the living part of a cell that is surrounded by a plasma membrane. Here, if I have a cell, then this is a cell membrane that I have. All kind of cell have a cell membrane first, and inside this there is cytoplasm, and inside nucleus there is nucleoplasm. These cytoplasm and nucleoplasm together known as protoplasm, and this word protoplasm was first given by J. Purkinje. Protoplasm contains the genetic material of a cell. It also controls the activity of the cell. Okay, and these are scientists that are there in front of your screen. The coined the word protoplasm, living material in the cell. It is by Purkinje. And what has Dujardin done? Said that cells contain a gelatinous fluid, a life substance, and they name it Charcot. He doesn't name it protoplasm, but he named it Charcot. He was Dujardin who first named it Charcot, and this name protoplasm was coined first by Purkinje, and he named it living material in the cell. Then there are Schleiden and Swan. What has they done? They have actually given cell theory. And what does this cell theory suggest? All living things are composed of one or more cells. The cell is the basic unit of life, and new cells arise from existing cell. This is a cell theory explained by these two scientists, Schleiden and Swan. That you can see, these are two scientists in front of you who has given cell theory. Then we have Rudolf Virchow. What has this scientist done? Virchow is credited with several key discoveries. His most widely known scientific contribution is he has also given cell theory. And what does his cell theory suggest? His cell theory suggests omnicellular e-cellular. Omnicellular e-cellular. What does this mean? Omnicellular e-cellular. It means all cells come from pre-existing cells. All cells come from pre-existing cells. That actually means omnicellular e-cellular. This statement was actually published by this scientist Virchow. He is also famous for explaining cell theory. Okay, the first cell theory was given by this Schleiden and Swan, and then second theory in 1855 was given by this Virchow. All cells come from pre-existing cell. It is very famous theory, and this one is also very famous. All living things are composed of one or more cells. He suggested about unicellular and multicellular, and also suggested that new cells arise from existing cell. But he suggested that all cells come from pre-existing cells. Then we have Robert Brown, another scientist who explained about nucleus. He is a Scottish and botanist, and best known for his contribution of cell's nucleus and of the continuous motion of minute particles in solution, which came to be called Brownian motion. He has explained Brownian motion. You must have heard earlier zigzag random motion. Robert Brown saw central structure in plant cells and called this structure as nucleus. Then we have Camillo Golgi, very famous Golgi apparatus was actually discovered in 1898 by this very famous physics physician. During an investigation of nervous system, he actually discovered Golgi apparatus. The Golgi body, I will tell you what the Golgi body does in our cell. The Golgi body is an organelle found in the most eukaryotic cell. It is found actually in eukaryotic. I've already told you what does prokaryotic and eukaryotic do and what is difference in these cells. Can you tell me what are differences in prokaryotic and eukaryotic cell, and what are differences in unicellular and multicellular cell that I have told you in previous class? I want you to tell me. Yeah. Okay. I have told you about those scientists because I was telling that Antony Leeuwenhoek was the first scientist who discovered free living cell, and bacteria and protozoa are actually free living cell. And Leeuwenhoek made microscope consisting of a single high quality lens to discover it. And then J. E. Purkinje first introduced the term protoplasm, and protoplasm is nothing but living part of the cell that is surrounded by plasma membrane. Then you can just quickly revise cell. That cell is the fundamental unit of life. All living organisms are made up of cell. It is the structural and functional unit of life, because all the life, all the body is made up of cell. It is known as the fundamental unit of life because it regulates all the functions inside an organism. And cell in Latin means a little room, and I have already told that when he discovered and observed the cell, he found that it looks like a honeycomb and all those compartments are so little. That's why he named it cell, that in Latin means a little room. And 1665 Robert Hooke does this. Then unicellular, multicellular, prokaryotic, eukaryotic. Okay, Sanket, I want you to tell me what you understand by unicellular and multicellular. Just present some point to differentiate between unicellular and multicellular. And also present some point to differentiate between prokaryotic and eukaryotic. Do that. Yeah, first tell me unicellular and multicellular, and then explain about prokaryotic and eukaryotic. You do that. Yeah, exactly. Here in unicellular, one cell is there, and that single cell does all structural and functional work. And here in multicellular, many cells are there. Okay, very good. The second point you can present that this single cell does all functional and structural work, and here many cells are involved. And in third point you can present example, isn't it? What are the example of unicellular organisms? Yeah, exactly. Amoeba is there, bacteria is there, Paramecium, Euglena, Chlamydomonas, all these are actually unicellular organism. And amoeba is very famous for their false legs, which are known as pseudopodia. And by using this pseudopodia, they do the egation, everything. Okay. And this Paramecium is quite like our slippers, and this Euglena is greenish uh, shape that you can see. Then what are different examples of multicellular? Can you tell me that? Yeah, exactly. All these humans, all these plants, they all are example of multicellular because they have many cells to perform those functional work and those activities responsible for survival. All right. And then we have prokaryotic and eukaryotic. Can you tell me, can you differentiate between prokaryotic and eukaryotic? Can you tell me? Can you tell me meaning of pro and karyotic? Can you tell me meaning of e and karyotic? First tell me meaning. Yeah. Yeah, exactly. Pro actually means before or primitive. Yeah, that is very old. And karyotic means actually it's about nuclear membrane or nucleus. Yeah, nucleus. That means if nucleus is old kind, it is prokaryotic. If true nucleus is there, R2, it is eukaryotic. When do you say a nucleus is true? When it has well developed nuclear membrane, you call it true nucleus. And if nuclear membrane are not well developed, you call it uh, poorly defined nucleus. In case of prokaryotic, nucleus is poorly defined, and here nucleus is truly defined. This is the first point. Here nuclear membrane is absent. Nuclear membrane is absent, and here nuclear membrane is present. And mostly unicellular are prokaryotic. And here multicellular are generally eukaryotic. Okay, I have differences here for you. Most prokaryotics are unicellular. Most eukaryotes are multicellular. The nucleus is poorly defined due to absence of nuclear membrane. The nucleus is well defined and is surrounded by nuclear membrane. Okay, then nucleolus is absent here. Nucleolus is present here. Cell organelles like plastid, mitochondria, Golgi apparatus, they all are absent here in prokaryotic. But these cell organelles like plastid, mitochondria, Golgi bodies, they all are present in multicellular. The examples of unicellular are bacteria, blue-green algae. Okay, and the examples of eukaryotes are fungi, plants, animal cells. Okay, got that? You can see the examples of prokaryote and eukaryote here. The prokaryotes are generally single cell. They are having single cell. Their uh, nucleus are not truly refined because they don't have nuclear membrane. In case of multicellular or eukaryotes, the nucleus is well defined, and all other uh, well defined body organelles, I should say, like Golgi apparatus, mitochondria, all these are present. Okay, then we need to understand about animal cell and plant cell. We all know that these two kind of cell are actually multicellular. If we consider an example of animal cell, they are multicellular. If we consider an example of plant cell, they again are multicellular. In these two, there are some similarities and there are some differences. Can you tell me what are similarities, Sanket, and what are differences? Yeah, between animal cell and plant cell. What are similarities that you find and what are differences? Okay. Yeah, and they both have nucleus, they both have cytoplasm, they both have plasma membrane, they both have Golgi apparatus, they both have mitochondria, they both have endoplasmic reticulum, they both have ribosomes. These are some organelles that they both have, and they are said to be similarities between these two. And if we differentiate between animal cell and plant cell, we can differentiate with the head, like we can differentiate with the shape as our one of the point. How can we differentiate from shape? If we differentiate with the shape, we can say that animals do not have fixed shape. Animal cell do not have fixed shape, but plant cell have fixed shape. The second point is on the basis of cell wall. How will you define on the basis of cell wall? Cell wall are not present in animal, but they are present in plant. Then how will you define them or differentiate them using vacuoles? Actually, vacuoles are very large in case of plant cells, and by using these vacuoles, they remove their waste. In case of animal, there is either no vacuoles, or if they are, they must be very small. Chloroplasts are not present in animal cell, but they are present in plant cell, and because of the chloroplast, they have a pigment, green pigment inside this known as chlorophyll, and because of this they can photosynthesize. That means they can prepare their own food, and this process can be chemically formalized like this. We can write the chemical formula in this way. They take carbon dioxide and water in the presence of sunlight and chlorophyll, and they give us energy. They give us glucose form of energy and oxygen. It's a very essential part that they take carbon dioxide and give us oxygen and some water vapor. You can just balance this equation known as chemical equation of photosynthesis. 6 CO2 plus 12 H2O in the presence of sunlight and the chlorophyll give us glucose, that is C6H12O6, and give us oxygen 6O2 and give us water 6H2O. And this is possible there in plant only. The plant only can photosynthesize or perform photosynthesis, and these animals can't do that. Now the question arises, how do they store the energy? The animal store the energy in the form of glycogen, but plant store it in the form of starch. This is the fifth point using which you can differentiate their activity. And then the last point is centrioles. Centrioles are present only in animal, but they are not present in plants. And in these 6 heads, you can differentiate their functionality. Got that? Then we have the structure of animal and plants. It's 3D structure. You can observe they look like this. It is a circular form, it is a rectangular form, and these are some parts. I will show you 2D structure because it is important to observe them in 2D. In 2D structure, they both have cell membrane, but in plant there is one more membrane here, and this membrane is actually known as you call it cell wall. And after cell wall there is cell membrane as well, that is plasma membrane, you can name it. Here we have plasma membrane, cell membrane, then we have different organelles, we call it cell organelles, like mitochondria, ribosomes, these small particles. Then nucleus is there, then nucleolus and endoplasmic reticulum, and cytoplasm and nucleoplasm inside nucleus, Golgi apparatus like here, and vacuoles, small vacuoles. Here large vacuole are there, and here we have again all these apparatus, Golgi apparatus, mitochondria, chloroplast, and these endoplasmic reticulum, ribosome, nucleus. But vacuoles are a bit larger here in the animal cell. I want you to draw these diagrams in your examination about animal cell and plant cell. Then we have different cell organelle, and these labeling that I have shown you: plasma membrane, cell wall, mitochondria. I will now tell you all these about all these cell organelle and their function. First is plasma membrane. It is the outermost covering, and it just separates the content of cell and outer atmosphere, external environment, and it looks like this. There are some protein and all these kinds of things. It is actually made up of lipid and protein, and its function is the selective permeability. It follow and it allow limited substance to go in and out, doesn't allow everything to go inside. It performs diffusion and osmosis for the same. Some substance like CO2 and O2 gas can move across the cell membrane by the process of diffusion. Diffusion is for gases and osmosis is for liquid. From high concentration to low concentration, this diffusion occurs. Then we have osmosis, the movement of water through such a selective permeable membrane, that is plasma membrane, is known as osmosis. And this osmosis perform these three kind of things you can observe: hypertonic, isotonic, and hypotonic. In hypo, the O for out, outside there is more water. In hyper, inside there is more water. If inside there is more water, water move from inside to outside, and that is why cell will shrink if you will just put them in hypertonic solution. If you will put a cell in hypotonic solution, as the water is more in outside, uh, the water move from outside to inside, that is why cell will swell. Okay, here cell shrink. And higher water, I'm saying that water is more inside, that means water will be less outside, solute will be more outside. Okay, there are two things here, water, that is solvent, and solute. Okay, and this process of hypertonic is also known as plasmolysis, and this hypotonic process is also known as deplasmolysis. Okay, what happens in plasmolysis? Losses water through osmosis. There is shrinkage, and here there is, I should say, in first case it shrink, in second case it swell. Swelling process is known as deplasmolysis. Then we have another cell wall, only present in plant cell. They are non-living, thick, freely permeable covering made up of cellulose, and cell membrane are made up of lipids and proteins. They are made up of cellulose. The function is determine the shape, rigidity, helps the transport of various substance in and out of the cell. Then we have this kind of cell wall you can observe, and then you can also observe cell structure. Briefly, you can just go through this. That cell are made up of component called cell organelles. A cell is capable to live and perform all their respective functions due to the presence of cell organelle. There are a variety of functions they are performed because of the cell organelle. All kind of cell organelle have specific function that they perform, and together an organism perform different activities because of this cell organelle. The structure seen in almost every cell is same. What are those same structure? Plasma membrane are there in every cell. Nucleus is there in every cell. Cytoplasm are there in every cell, and then other cell organelle may vary. Then cell wall are found only in plant cell, and they are made up of, I have already told, they are made up of cellulose. Okay, cell wall are actually made up of, I have already told you, cellulose. Then they are rigid covering, and then plasma membrane and its function, you have already understood that what the function plasma membrane has, and they are made up of lipid and protein, and they perform uh, osmosis and diffusion to transport gases and liquids. Then we have cytoplasm, a liquid inside a cell, thick solution composed of water, salt, protein, and it is surrounded by cell membrane. Nucleus of the cell is surrounded by the cytoplasm. That means there is a cell membrane, then there is a nuclear membrane, and the liquid is filled between cell membrane and nuclear membrane is actually cytoplasm. And the liquid filled inside this nuclear membrane is nucleoplasm. You can just write it. And these two liquid together are known as protoplasm, and the name was given by Purkinje, if you remember. Got it? Then we have nucleus. Nucleus is dense and spherical organelle. It was discovered by Robert Brown. Okay, I have already told you that who are those scientists: Robert Hooke, Purkinje, Robert Brown, Sheldon Swan, and different scientists. I will even help you with them once again later on. Nucleus is bounded by two membranes, both forming nuclear envelope. Nuclear envelope contain many pores known as nuclear pores, and nucleoplasm is liquid inside that nuclear membrane. Then the functions, it is known as brain of the cell because nucleus control all the metabolic activities. Very important point. It regulates the cell cycle. It is also known as storehouse of the gene because what we inherit from our parents is decided by the gene material present inside this nucleus. That is why we call it storehouse of genes. These are different names given to this: brain of the cell, storehouse of the gene, and transmission of hereditary traits from parents to offspring is decided by these genes. Then we have cytoplasm. I have already told you that protoplasm is composed of the cytoplasm and nucleoplasm. This is jelly-like viscous colorless semi-fluid, and plasma membrane and nuclear membrane. And between plasma membrane and nuclear membrane, it is present. Then cytoplasm acts as a store of vital chemicals. What are these chemicals? They are amino acid, they are protein, they are sugar, they are vitamins, and we call it brain of the cell, and we call it storehouse of the gene. Then we have this basic structure: cell membrane, nucleus. Here we have cytoplasm. Between cell membrane and nuclear membrane, you can observe it from this diagram. Then we have one more cell organelle known as Golgi body or Golgi apparatus. It was discovered by this scientist Camillo Golgi in 1898 and named after this scientist Golgi. Golgi apparatus discovered in 1898 by Italian physician Camillo Golgi during an investigation. He was investigating about nervous system, and during that investigation he found this apparatus. And the sacs or folds of the Golgi apparatus, these sacs that you find here, this kind of sacs are here. What do you call to these sacs? These sacs are known as cisternae. Then in 1873, Camillo Golgi created the black reaction technique, which enabled scientists to stain and view the structure of neuron, their specialized cell. He also discussed all those about nervous system, neuron. His main discovery was about nervous system, and during that observation, he actually discovered this Golgi apparatus as well. In this technique, cells are stained using potassium dichromate and silver nitrate. In 1873, Camillo Golgi used the chemical potassium dichromate to harden the tissue. Golgi received the highest honors and awards in recognition of his work. He shared the Nobel Prize for 1906 with Santiago Ramon y Cajal for their work on the structure of the nervous system. He has even received the Nobel Prize, the highest honor for his discovery of nervous system. What are the functions of this Golgi apparatus? It is cell's post office. What it is? It is cell's post office. What do we call this? Post office. Because in post office we sort out address and then transport. Same thing is done by this Golgi apparatus. A major function is the modifying, sorting and packaging of proteins. What do they do? They sort, they package and they modify these proteins for secretion. It is also involved in the transport of lipids around the cell, and they also form the lysosome, which are suicidal bag of the cell. These are different functions of this Golgi apparatus. Then I have already told you that lysosomes are formed by these Golgi apparatus. Now the question arises what these lysosomes are. They are again cell organelle, and these are very small spherical sac-like structure. They contain several digestive enzymes, and that is why they can digest any foreign particles They are found in eukaryotic cell, mostly in animals, and the lysosome has the function of digestion of foreign substance. That means if some uh, external particles come in contact with these cells, they digest them. During the disturbance in cellular metabolism, for example, when the cells get damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, lysosomes are known as suicidal bag of the cell. Why do we call them suicidal bag of the cell? Lysosomes are suicidal bag of the cell. The question may be there, which uh, storehouse of the gene? Can you tell me which cell organelle is a storehouse of the gene? Storehouse, not powerhouse. Yeah, powerhouse of the cell. Storehouse of the gene, I'm saying. Genetic material, I'm saying. Yeah. Cell's post office. These are very interesting thing that you should know. Cell's post office. Yeah, cell's post office is Golgi apparatus. We have just studied. Yeah, I have just told that as well. The storehouse of the gene. It is nucleus. Yeah, it is nucleus. Powerhouse of the cell is correct, mitochondria. Okay, I'm presenting them all together so that you can remember. Okay, lysosome is suicidal bag of the cell. Or simply you call it suicidal bag. What they are, lysosomes they are. Okay. Okay, it is L. Okay, all right. Then we have these digest the foreign substance, suicidal bags of the cell. Then endoplasmic reticulum are just attached to this nuclear membrane like this. They are smooth endoplasmic reticulum if they don't have ribosome, and they are rough endoplasmic reticulum if they have ribosome around them. It is membranous network of tube-like structure extending from nuclear membrane to plasma membrane, and it is absent in prokaryotic and matured RBCs of mammals, and they are like this. And there are two type of endoplasmic reticulum, and their main function is synthesis of protein, and they also transport this protein. And it helps in transport of various substance, and it acts as a scooty, you can say. And RER, that is rough endoplasmic reticulum, help in synthesis and transport of protein, and SER help in synthesis and transport of lipid. These are two, smooth endoplasmic reticulum and rough endoplasmic reticulum. This SER will work for lipid. It will synthesize lipid and transport lipid, and rough endoplasmic reticulum has ribosome, and ribosome synthesize protein and just transport it. Got it? Yeah, then we have ribosome. I've already told you that it helps in synthesis of protein, major role in the synthesis of protein, and these are made up of RNA, and these are extremely small, dense and spherical bodies which occur freely in the matrix or remain attached to the endoplasmic reticulum. And other name is protein factory. What do we call to this? Protein factory we call to this. Now I want you to write about this protein factory. Here I was writing it. Post office of the, yeah, here I was writing protein factory. If someone asks, then what will you, which organelle is protein factory? Tell me. Ribosomes are. Oh, ribosome. Okay. Now just focus here. The storehouse of the gene, nucleus. Powerhouse of the cell, mitochondria. Cell's post office, Golgi apparatus. Suicidal bag, uh, lysosomes. Protein factory, ribosome. What is the function of RER? It synthesizes protein because and transport protein. What is function of SER? It synthesizes lipid and transport lipid. Then we have this kind of ribosome around these rough endoplasmic reticulum. Free ribosomes are here, rough endoplasmic reticulum. This is protein factory. Then we have mitochondria, also called as shoe-shaped organelle, and they are double membrane structure, with outer membrane being smooth and porous, whereas inner membrane being thrown into a number of folds. Inner membrane has many folds, and upper membrane has shoe shape, or you can say the smooth porous. And they contain their own DNA and ribosome. They have their own DNA and ribosome. And mitochondria store the energy. That is why they are powerhouse of the cell. They are the sites of cellular respiration, and due to this cellular respiration there occur release of energy, hence provide energy for the vital activities of living cells. They store this energy in the form of ATP, known as adenosine triphosphate, and energy currency of the cell, or powerhouse of the cell. Powerhouse of the cell, I have already written there, powerhouse of the cell, or energy currency of the cell. Powerhouse of the cell, or energy currency of the cell. You can just write it, energy currency of the cell. And energy currency, what is the energy currency? This is adenosine triphosphate. It is stored as ATP. Then after mitochondria that we have discussed, I want you to go through these quick revision. Nucleus, nucleo double layer, all these things. Golgi apparatus, quickly go through this. Front fish Cameo Golgi discovers, store, modify and package cell product. Complex sugar are made from simple sugar in the Golgi apparatus. It also involves the formation of lysosome. Then lysosomes, suicidal bags they are. It is a waste disposal system of the cell, and this is powerful digestive enzymes are present. RER and RER and SER I have to explain. Then mitochondria I have already explained, they are powerhouse of the cell, and DNA, therefore they can make their own protein. They have their own ribosome. Then we have plastids. Plastids are present in most of the plant cell and absent in animal cell. I have already explained this, that plastids are absent in animal cells. They are usually spherical or discoidal in shape and double membrane bound organelle. They also have their own DNA and ribosomes. And the plastids that I want you to understand, there are three kind of plastids: chloroplast, chromoplast and leucoplast. These are three kind of plastid: chloroplast, chromoplast and leucoplast. Chloroplast is for green, chlorophyll. Inside chloroplast there is chlorophyll. Green pigment it is. Chromoplast, colored other than green. Okay, you can read from this point as well. Chloroplast, these are the green colored plastids, contain chlorophyll, and that is why chlorophyll is responsible for photosynthesis present there, and that is why plants which has chloroplast can photosynthesize and can prepare their own food. And there is chromoplast is something that has color other than green. These are colorful plastid. Okay, chloroplast is green plastid, green colored plastid. Chromoplast is colored plastid, and color is other than green. And leucoplast is colorless plastid. These are difference in these three kind of plastid. Now the question arises, what are the function? I have already told you. Chloroplast trap solar energy and utilize it to manufacture food for the plant. That means they can manufacture food for the plant. This is the function. Then this chromoplast impart various colors to flowers. Various colors can also attract insects for pollination, and leucoplast may also perform biosynthetic functions such as the synthesis of fatty acid, amino acid and various other compounds like storage of starch, lipid flow, protein. And these plastids, I have already told that double membrane bound structures they have, present in plant cell only, have their own DNA and ribosomes, and they are enfolding of membrane, and this is called as matrix, called as thylakoids and matrix. It's called stroma, seat for enzymatic actions, and these are chromoplast, leucoplast, and chloroplast, and colored plastid is chromoplast, and leucoplast is actually colorless. It stores materials such as oils, protein, fats, and green pigment chlorophyll is present, which is helpful for photosynthesis. Then we have different color. You can observe that what is the reason behind color of your uh, tomato. Maybe what is the reason behind, what is the plastid involved for color of carrot, what is plastid involved for color of other fruits. You just go through this, and I will you just search all these points that I have just asked. Then we have vacuoles. Vacuoles are liquid, solid, filled and membrane bound organelle. In plant cell, vacuoles are large and permanent. In animals, they are so small and temporarily. In mature plant cell, it occupies 90% space. What is the function? They perform they help in maintaining the osmotic pressure in cell. They provide turgidity and rigidity to plant cell. This is the function of vacuole. Then I have just told who the Robert Hooke is and what has he done. Same I have explained about Anthony von Leeuwenhoek. Then I have told you what has Purkinje and Dujardin done. And Seldin and so on has given cell theory, and that cell theory explains this thing, that all living things are composed of one or more cell, and the cell is basic unit of life, and new cell arise from the existing cell. And there is Virchow, who has again given the cell theory, that means omnicellular e cellula, which means all cells come from pre-existing cell. Then we have Robert Brown, who has just discovered nucleus and also the Brownian motion. And then Camillo Golgi, I have already told you that in 1898, when he was working on nervous system, for which he has also received Nobel Prize, during that uh, task observation, he uh, randomly discovered the apparatus responsible for transport, manufacturing, uh, quite like post office, packaging work, and sorting work is done by this apparatus known as Golgi apparatus. Then we have this named after him, discovered in 1898. Now we have some questions for you. Quickly go through them, and then we will end the session for today. Don't worry. Okay, we have some questions, and I want you to quickly go through this. Cell is a Latin word. What does it mean? I have four options, and I have already ticked here. A little room is the meaning of this cell. Then the second question that I have is plant cell contains. What do you think? What does they contain? In all plant cell we have, yeah, everything, because in plant cell we have chloroplast, inside that we have chlorophyll. Cell wall they have, nucleus they have, plastid they have. In fact, chlorophyll is chloroplast is a plastid. Okay. Then we have plasma membrane is a selective membrane, while a cell wall is fully permeable. Okay, cell wall is fully permeable, but cell membrane is semi-permeable. Then we have carbon dioxide moves out of the cell by the process. Obviously, gas has a process of diffusion. Then we have a plasma membrane consists of. What does they consist of? Chitin or cellulose or protein and lipids or fibers. The answer is cell wall is made up of cellulose, cell membrane is made up of protein and lipids. That means protein and lipids is your answer. Then the fundamental unit of life. You know the fundamental unit of life is obviously cell. Fundamental unit of life is cell. Now these are some questions. Who discovered cell and how? You already know that. Why is the cell called the structure and functional unit of life? You know this as well. How do substances like CO2 and water move? The process is diffusion. And why is the plasma membrane called selective permeable? Because it allow only selective component to come in and come out of the inside cell and outside. Who discovered cells and how? The reason you have already understood. You have already gone through this. Okay, why is this plasma membrane called selective permeable? I have already explained. Prokaryotic and eukaryotic, you can just go through this and fill this. Here I have filled even the size and there are these differences. Then we have, can you name the two organelle we have studied that contains their own genetic material? Mitochondria has their own genetic material. That's why they can, uh, yeah, they can create their own protein as well because they have ribosome as well. Plastid has their own genetic material. Then if the organization of a cell is destroyed due to so physical or chemical influence, what will happen? Suicidal vac, you will explain here. Why are lysosomes known as suicidal vac? You know the reason. Where are proteins synthesized inside the cell? Ribosome. Then make comparison between plant and animal. You can compare. I have already told all these things. How is prokaryotic cell different from eukaryotic? You can just go through this. Then what would happen if the plasma membrane rupture? You will not be able to separate uh, all organelles from outer environment. What would happen to the life of the cell if there was no Golgi apparatus? Actually, Golgi apparatus store, package and manufacture substances, and this will not be done. Cell will not be produced actually. Then which organelle is known as the powerhouse of the cell? Mitochondria. The reason you can understand. And then lipids and protein consisting the cell membrane get synthesized, where the answer is endoplasmic reticulum. And in smooth, yeah, there are two kind of endoplasmic reticulum, SER and RER. In smooth endoplasmic reticulum, there is the lipid formation and synthesis and transportation. In RER, there is the protein synthesized and transported. Okay, you remember that. How does an amoeba obtain its food? You can just understand the pseudopodia, the false legs they have, through which they can just get their food. They can also excrete waste material from this pseudopodia itself. Then there are some experimental activity, and the last question is based on this cell division. Okay, I think you are okay with this explanation. We have almost